Hello everyone, this is the group presentation for the course Biocomputation and Bioinformatics, and our title is Comparison between Cutinase 31174, Thermobifida fusca bacteria, and Fusarium vanettenii fungi. Our group members are Farid Nahyuddin bin Mohanazuddin, Nazliani Amidia binti Nurjantai, Nurhanis Adwa binti Hasrizal, and Sofia Anak Ngaden. For the introduction, we have decided to choose Cutinase 31174 as our enzyme to make the comparison. Cutinase are enzymes that belong to the alpha-beta hydrolase superfamily and are characterized by a catalytic triad composed of SHAHIR, HIS, and ABS. These enzymes are known for their ability to catalyze a wide range of reactions involve various substrates including short-chain soluble ester, water-insoluble triaglycerol, polyester, and wax. The comparison of cutinase from two distinct sources, which are Thermobifida fusca, a bacteria species, and Fusarium vanettenii, a fungus species. Thermobifida fusca, a thermophilic actinobacterium, has evolved its cutinase to operate efficiently at high temperature, a characteristic that aligns with its evolutionary background in hot, compost-like setting. This thermostable enzyme not only aids in the degradation of cutin, but also demonstrates exceptional stability and efficacy over a wide temperature range, positioning it as a promising candidate for diverse industrial and biotechnology applications. Fusarium vanettenii, a versatile fungus species, has developed a cutinase variant that functions optimally under mesophilic conditions. This enzyme is pivotal in the fungus' capacity to colonize and break down plant material, contribute to its ecology success as a saprophyte and potential plant pathogen. And then we use Brenda at the early stage of the study. Brenda is a comprehensive database of information on enzymes, include their properties, function, and role in various biological processes. It is widely used by researchers in biochemistry, molecular biology, and related fields. Then, our main research question is, what are the key structural and functional differences between the cutinase enzyme from the thermophilic bacterium, which is Thermobifida fusca, and the mesophilic fungus Fusarium vanettenii, and how do these differences contribute to their distinct catalytic properties, substrate specificities, and potential application. Problem statement. Our current understanding of enzymatic properties, stability, and substrate specificity across different biological sources remain limited. There is a significant lack of comparative data on the enzymatic kinetic, stability, and substrate specificity of cutinase from bacteria and fungi. There is insufficient structural characterization to understand the molecular basis of functional differences and the potential application of diverse cutinase in new and existing biotechnological processes are underexplored. So our research aim is to propose a comprehensive study of cutinase through comparative analysis derived from Thermobifida fusca and Fusarium vanettenii by explore their enzymatic properties, structural differences, and also functional capabilities. And our research goal are contribute to a deeper understanding of the comparative enzymatic properties, structural insight, and application potential of cutinase enzyme between Thermobifida fusca and also Fusarium vanettenii. And last one is to provide valuable insight that can enhance the application of cutinase in various biotechnological fields. We have three objectives, which are Objective 1, to perform a structural and functional comparison between the cutinase from Thermobifida fusca and Fusarium vanettenii. Objective 2, to identify the key structural features that are responsible for the differences in catalytic activity and substrate specificity. And last one is Objective 3, to explore the potential application of this cutinase based on their unique properties. Scope of study. A comprehensive computational analysis and comparison of cutinase from two distinct sources, Thermobifida fusca, a bacterial species, and Fusarium vanettenii, a fungus species. Employ a bioinformatic tool and technique to investigate the structural and functional aspect of this enzyme. The study will involve sequence analysis, multiple sequence alignment, and phylogenetic analysis to predict and compare the 3D structure of the cutinase from the two sources. Various informatics databases and software will be utilized for functional annotation, structural analysis, and identification of critical region or residue that responsible for potential differences in catalytic activity and substrate specificity. Significance of study. This research aims to provide insight into the evolutionary divergence and adaptation of cutinase to different environments. Understanding the structural basis can facilitate the rational design and engineering of cutinase with desired properties tailored for specific applications such as biofuel production, biodegradation of polyester, or textile processing. The implications of this comparative study are multifaceted, including the identification of unique structural features that confer enhanced catalytic activity, stability, or substrate specificity to bacterial or fungal cutinase. It can shed light on the evolutionary adaptation of this enzyme to diverse environment and substrate, guiding the development of tailored cutinase variant through protein engineering or directed evolution approaches. This research will contribute to the knowledge base of cutinase, diversity and structure-function relationship, paving the way for future research and application across various fields. For the literature review, these are the industrial application of cutinase that has been done in the past. First, textile sector was the first to use industrial cutinase because it can aid in cotton scouring, which increase the water resistance of cotton fiber. And then cutinase can be used in the processing of leather and synthetic fiber, taking the place of conventional physical and chemical procedure and optimize the process while lowering expenses and energy usage. And then decolorization of wastewater made from molasses in sugar mill. The decolorization rate may reach 80%. And then cutinase can improve the effectiveness of pesticide to raise agricultural yield and lower pesticide usage. The food sector also use cutinase to process dairy product, flavoring, and dehydrated fruit. Cutinase perform effectively. And then cutinase can be applied as laundry and dishwashing detergent. And lastly, cutinase can be applied in the hydrolysis of paper making, sticky during the paper making process.
Moving on to our selected fungi, which is Fusarium vanettenii, or also known as Neocosmospora pc. These fungi have Cat1 gene and contain 230 sequence of amino acids. From UniProt database, we can analyze the specific protein sequence that we will be used to perform the comparison of a structural and functional with selected bacteria. Here are the 230 sequence of amino acid of Fusarium vanettenii that we will use for fungi bacteria comparison. Next is the active site of the Fusarium vanettenii in the amino acid sequence. Based on the diagram, the alphabet with red square and dot indicates the active site of Fusarium vanettenii. In here, we could analyze there are some active sites present in this fungi. Next is blast result of Fusarium vanettenii. We perform the blast of the selected fungi by using UniProt database. So, here are the top 10 blast results for Fusarium vanettenii. There are Fusarium vanettenii, Fusarium solani subspecies cucurbitae, Fusarium solani or filamentous fungi, Fusarium falciforme, Dactylonectria stenocrastodontis, Fusarium duplospermum, Inorceratia species, Curatia gorensis, and Fusarium piperis. Next is the phylogeny tree of Fusarium vanettenii. This phylogeny tree represents the evolutionary relationship among the top 10 blast results for the Fusarium vanettenii that we obtain from the UniProt database. The analysis was performed using sequence alignment and phylogenetic tree construct method. First, we performed the sequence alignment of Fusarium vanettenii by using the UniProt KB. The top 10 blast result for the fungi then aligned to identify the similarities and differences among them. Next, we will use the molecular evolutionary genetic analysis, or also known as MEGA. The aligned sequence were then used to construct a phylogenetic tree using the MEGA tool. MEGA provides the various method for phylogenetic analysis, and the tree was generated using the neighbor joining method with the bootstrap values to indicate the confidence levels of the branches. So here is the result of the phylogenetic tree of the Fusarium vanettenii.
Let's move on to our chosen bacterium, Thermobifida fusca, which has 319 amino acid sequences. By utilizing the UniProt database, we can analyze the specific protein sequence needed to compare structural and functional aspects with selected bacteria. Here are the 319 amino acid sequences of Thermobifida fusca that we will use for fungi-bacteria comparisons. The next focus is the active sites within these amino acid sequences. In the diagram, the latest marked with red squares and dots indicate their active sites of Thermobifida fusca, showing that several active sites are present in this bacteria, while the black squares in this slide indicate their binding sites. Next, we have the blast results for Thermobifida fusca. The blast analysis was performed using the UniProt database, and here are the top 10 blast results for Thermobifida fusca, including Thermobifida fusca strain YX, Thermobifida fusca itself, Thermobifida cellulosilytica, Thermobifida cellulosilytica TB100, Thermobifida alba, and Thermobifida halotolerans. Despite some organisms having the same name, they differ in their gene names. Moving on to the phylogenetic tree of Thermobifida fusca, this tree illustrates the evolutionary relationships among the top 10 blast results. The analysis was conducted using sequence alignment and phylogenetic tree construction tools. First, we perform a sequence alignment of Thermobifida fusca using the top 10 blast results obtained from the UniProt KB database. The sequences were aligned to identify similarities and differences. Then, we used the MEGA tool to construct the phylogenetic tree. MEGA offers various methods for phylogenetic analysis, and in this case, the tree was generated using the neighbor joining method with 1000 bootstrap values to indicate the confidence levels of the branches. So here are the results of the phylogenetic tree for Thermobifida fusca.
Today, I will be presenting a comparative analysis of protein structures from two different species, Thermobifida fusca and Fusarium vanettenii. The analysis includes sequence alignment, active and binding sites visualization, 3D structural representations, and an alignment of the 3D structures to highlight structural differences. So let's start with the sequence alignment. Here, I have displayed this sequence alignment between Thermobifida fusca and Fusarium vanettenii, which I obtained from the pairwise alignment tool provided by the protein data bank. This alignment helped us to understand the similarities and differences at the sequence level between the two species. Next, I present two images. The first image shows the active site between Thermobifida fusca and Fusarium vanettenii. This comparison reveals the functional regions that are conserved or differ between the two proteins. The second image indicates the binding sites of these two proteins. Binding sites are crucial for the protein's interaction with other molecules that can provide us insights into their functional similarities and differences. Moving on to the 3D structures. For Fusarium vanettenii, I use PyMo to generate the 3D structure. In this structure, the active sites are highlighted in yellow and the binding sites are colored in magenta. This visual representation helps us to easily identify these critical regions within the protein structure and For Thermobifida fusca, I obtained the 3D structure from the protein data bank. In this model, I have highlighted the binding sites in blue and the disulfide bridge in red. The disulfide bridge are important for the stability and folding of the proteins. Finally, I will discuss the alignment of the 3D structure between Thermobifida fusca and Fusarium vanettenii. The structural alignment indicates a slightly difference in shape between the two proteins. This difference could be due to variation in their sequences or the presence of unique structural elements, which might affect their functions and interaction within other molecules. So, in conclusion, this study compared Cutinase enzymes from Thermobifida fusca, a bacterium, and Fusarium vanettenii, a fungus, focusing on their structural and functional characteristics. Our analysis revealed both similarities and significant differences between these enzymes. Both enzymes have the same number of active sites, but these sites occur at different positions in their amino acid sequence, leading to variation in the substrate binding regions. So these structural differences influence their catalytic activity and substrate specificity. Additional, Additionally, the 3D structures of the enzyme show significant differences in size and shape. Using the BLAST tool at the National Center for Biotechnology, NCBI, we examine the species similarity percentage and generate phylogenetic tree helps to analyze the evolutionary distances and connection between species. Overall, the versatility of Cutinase in substrate hydrolysis, its potential for various industrial applications, and ongoing research to enhance its efficiency and stability highlights its significance in biocatalysis and biotechnology. That's all from us. Thank you.